It's on now. Okay, hello, welcome back, welcome back. Please take your seats. Okay, welcome back everyone. Hope you enjoyed your lunch. It's my pleasure to introduce Professor Susan Holmes. She's barely needs an introduction to this audience, and so I'll keep this short. She's especially well known for her work in non-parametric multivariate statistics, does a lot of applied work in microbiome analysis, and also is co-author of this very well-known book, Modern Statistics for Modern Biology. And today she's going to be talking about latent variables for heterogeneous data.
Thank you very much for the invitation. It's a wonderful community to be part of, and it's one of the selling points of using Bioconductor is the community and the people who put all that energy, so thank you so much. I work on mathematics, biology, and statistics, and I try to build bridges. And I'm going to give you a little bit of a talk. Now I'm emeriti, so I can talk about whatever I like. It's, so you get to hear me say. I've been using various large language models for the last 2 years, and especially since I've deprived of a group of students, Claude is my graduate group. And I've encountered a lot of mistakes. And I'm not talking about hallucinations. I know how to use it very, very well. And I wanted to talk to you about things that I think humans should pay more attention to, because I think this is the winning points. It's the latent variables and identifiability problems that come up. And so in biological systems, there are, of course, very important structure that we don't measure. And very often, those are the things that we call the latent variables that we're going to try to understand better. And we have very high dimensional data, there's no doubt about that. And I'm going to start with what I'd call a silly example, but it's one of the first examples for me where I realized that there was a problem about alignment and what we call registration. So, but where do other examples of latent variables come in? Well, we all have pseudotime, or the severity of a disease, the progression, the development of a cell. Sometimes you have continuous multidimensional data, and you might even have discrete data. And those of you using large language models, for instance, know how important topics and tokens are, because they are the mixed membership models on which the representations of the data in these models. And so I'm going to talk about that. But one of the things I think, which is, why are the large language models so successful in some cases, and that has to do with context. And I think that you have to be very, very careful about what you do with your data so that you keep the context. And so the context allows you to give alignment and registration. And we use the context, we can use it in a Bayesian way, or you can have multi-table ways of looking at rotations and things like that. But there is, if you're lacking context, very often you come across what's called the identifiability problem. And that means that, so for instance, take a clustering, and you did one instance of the clustering, and you called the clusters 1, 2, 3, 4, and then another instance where the clusters don't have the same labels. We call that non-identifiability of the labels. And so if you have the same data and different latent representations, you lose the identification. And that loss can lead you to make your data more noisy than they are. And I'm going to give you my little example. So this is a study I did 20 years ago. And it was my first incursion into image analysis. And I was studying the dynamics of the coin toss. And I'm going to try, and I'm just going to bring this up and try again. It might come up. So those are the ellipses from the coin. As we flip the coin, and this is the original video of somebody flipping a coin with a very, very high-speed camera. This was very difficult to do at the time because we had to have a camera which was tied down to a table, and in order to have very, very high speed, it was a big installation at the engineering department at Stanford. But we were able to capture the speed of toss of the coin, and what we wanted to do is ask the question about if you toss a coin with the heads on the top, is it more likely that heads come up at the end? And this is about real people, not my husband, but real people, how they toss real coins. And so we were trying to understand the dynamical bias of the coin toss, and we did this using data, which was fit to a model, which was a physics model. And the images of data, as Wolfgang always likes to say. And here we have the ellipses, and this is how we fit the data with, edges, edge detection. It's pretty good. You get a series of ellipses, and we wanted to find out what the precession was in the various tosses of the coin in order to put it into our model. And so it's a pretty easy image analysis problem. It's ellipses and the projection of disks. And so you know what you want. And when we finished, when we computed the normals, what we get from our estimate of the ellipses was something like this. And so in this data, and I don't know whether I'm going to be able to point, probably I can. Here you see 128, 129, I don't know where 130, but there's 136, 105. And so these are actually the images, the sequence of the image. And in some sense, that's one of the latent variables. You have pseudo-time in some sense. We have time. And you can see a little bit that they're all over the place. You have 140 over there, 135 here. So the data are pretty, the data are pretty noisy. And so after doing registration, that is getting rid of some of the noise by aligning the data carefully, we get this. This image is much more satisfactory. And in this, in this image, we have here 119, 120, 121. You see the sequence is established. And so what happened? Well, the main thing that happened was in my equation here, I have 3 epsilons, which are signs, which could be one or the other. And they're not identifiable. That is, in my estimation, they could have been either way, and sometimes they're estimated one way, and sometimes they're estimated the other way. You will have seen this if you do principal components. Sometimes all the coefficients are positive, and then for practically the same data, you get them all negative. They're not identifiable. So you have to do registration, and that is one of the things that people forget to do when they're doing latent variables. And so this is labeling requires alignment or registration, and we have different ways of doing it. Multiple tables or multivariate angles are one way of doing it. You could do it in the Bayesian way and say, okay, the next normal mustn't be more than an angle of 90 degrees from the previous one, or you put it in a Bayesian model and do it with MCMC. But that's what we did when we reorganized the data. And the results from the movies, which I'm quite proud of, we had 50 different coin flips, and we made an estimate of about 0.508 of the probability of it coming heads. So there's a small bias in favor of the way you started. And recently, there's confirmation done by a team in Holland where they did 350,000 coin flips in a very, very well-designed experiment. And their results showed they got an estimate of 0.508 with a credible interval of 506 to 509. And this happened, this has been published in JASA this year, so I'm very proud of the data. And it doesn't have anything to do with biology, but it does have to do with this problem that we get all the time, which is registration and alignment. So other types of alignment have to do with when you're doing multiple omics, it's the same thing. You need anchors, and you need ways of doing registration. So if you're looking at microbiome data, and there are a lot of challenges. They're not homoscedastic, they're not IID, they don't sit necessarily on a manifold, and they're not predefined, so you can't define them as compositional. And in general, they come with also mass spec data and RNA-seq or other types of measurements, like we use a lot of MS metabolites now, and we have metabolic networks. So there's a lot of registration involved because you're trying to look at the data in a lot of different angles. So first of all, you have to denoise. And that is part of the package called Data2, where we denoise the data not by using a radius of fixed size, because that would be unfair to the points which you have a lot of instances of, but instead we do a probabilistic model. And in the probabilistic model, we just make it that the read goes to the group that it's more probable to belong to, and we can then model the noise. Every time we have a spelling error, we can model that, and that enhances the model. And the advantages of that is that you don't lose information. And one of the most important consequences is I study the vaginal microbiome of women in the context of HIV and various preterm pregnancies. And it's very important which strains of the bacteria are present, and we can see the strains, and in particular, this is Lactobacillus crispatus, which we use actually in clinical trials to improve the microbiome. But this has been a big breakthrough, was this denoising or registering de novo. So we make the features ourselves, so featurization in some sense. And this is the output, that is, you get a strain table of the different reads of different types. And so, for instance, these are what we call amplicon strain variants of different types, and then we have the samples. And so they're not fixed a priori. That is, you don't go in and say, I'm measuring, carbon, nitrogen, oxygen, as you would if you were really doing chemistry. Here, the data are, the categories are made from the data de novo. And that has consequences because you can detect if you have contamination, or you can also see various kinds of synthropies between the bacteria which are interacting. So you can see the interactions. But it's very important to remember the data themselves are not compositional. If you're going to make them into relative abundances because you want to, you're making a choice and you're losing information. So many times I have people come and see and they say, these are the relative abundances, and they don't know what the denominator was. And so they've lost all the information about the variance. So it's very important to keep all the information for yourself, going down the road. So the next challenge is interpretation, and ASVs, or different strains of bacteria, can't be considered as multinomials because they're not independent. So some bacteria need other bacteria to grow, others are they're antinomic, so you can't grow them together. And so it's very, you need a model which is better than the ordinary multinomial, and we use a Dirichlet mixed membership model, which was something which first was developed actually in genetics for the admixture models. And then it became very popular for language. And it's one of the ways in which tokens are made for the large language models. So it's a mixed membership model. Now in language, for instance, you go from, you have words, and the words come really from, they make terms, so you go up. And for us, we have reads, and the reads indicate the species by using denoising. And the species are assembled in communities, which are probabilistic membership models. And for topics in the document, you would, from the terms, you make a topic. And so within the topic, if you're doing sports, then maybe the field is a topic which occurs a lot. And so you can tell by the probability distributions at which topic you're in. And this is the same for the communities. And this is the latent Dirichlet model, or latent variable model, that we use the most for the microbiome. And so it's a Bayesian model, but you can also do it as a sort of hierarchical model. And the advantage is you keep all the information, so you keep things probabilistic. So in a topic model, a point is a measure, and it can be a little bit, strong on topic 1, but a little bit of topic 3 and a little bit of topic 2, and so it could be here. In clustering, there's a hard boundary, and you're either in one cluster or another, and it's discrete. Now, the hard boundary has a disadvantage because what we're looking at mostly is longitudinal data. We're trying to see transitions from states, and these transitions, you don't see the moment where it's going to occur when it's black and white. It is this hard threshold. If you have a soft threshold, you can see it happening better, so you have more information. So the statistical model that we have, this latent Dirichlet, is the standard topic model. And the output, here's the longitudinal data, which is patients taking antibiotics. And you see this first topic, which was many species had this profile in some sense. And here we didn't take into account the sequential structure. We just did every sample separately, but you can see that this first topic, about 3 days after when they were taking the first antibiotic, you have a big drop in all of those species. And the second time they took antibiotics, the drop happened slightly earlier, but it bounced back much faster. And then you have a second type of topic where there's much less of a drop, and the second time it goes up. So these sort of profiles, in some sense, or signatures, these are the topics that enable you to summarize and understand the data better. And so this is just the result from the topic analysis. And then the interpretation, sometimes it's difficult to know how many topics do you need. That's an alignment problem as well. So if you do it and you look at how, if it was separated in 2 topics, who belongs to the 2 topics? If it's separated in 3 topics, you can ask, which of the species are in this topic, and the alignment occurs between the first split and the second split, and then the second split to the third split. And you keep these up so that you can see what are the stable parts, what are the parts that stayed together, and you don't lose the labeling in some sense. So this alignment of topics. Now, this is a very nice package that was developed by 3 of my students and postdocs, Julia Fukuyama, Chris Sankaran, and Laura Simmel, and they have a nice paper in biostatistics, and ALTO is the R package, which enables you to see where you want to choose the number of topics. And again, this is done by doing this alignment. We're able to compare, for instance, in pregnant or non-pregnant subjects, the topics, and there's some topics which are sort of particularly bad. This is a Gardnerella-based topic, and we have it also in purple down here. We see it both in the non-pregnant and the pregnant subjects. This is a problematic topic, which is pathological. And so we have these assemblies of little communities of bacteria which co-occur. And in some sense, they don't always co-occur. Topics are very flexible, and so it's like in sentences, you can have synonyms, and one replaces the other in another context. And this is very important because sometimes you don't have that strain, but that strain's replaced by another strain which has the same function in a different population. So the topics enable you to cater to that. And when we're doing the bootstrap or Bayesian PCA, you have the problem that you repeat your principal component analysis many times, and you want to see the contours, how sure you are of the projection of the points. Now, these are nice output from a paper that came out with Lian Yuan and that we published in PLOS Computational Biology, but the original data, when we projected the principal components, the same point were projected very far apart. And I remember saying, that's just like the coins. That is, it hasn't been aligned. And once you align them, you realize that, in fact, in some of the data, the first and the second axes of your principal components have been switched, and the signs can be switched. And so you need to do the relabeling or the registration in order to have a projection which is neat, like these ones. So that's a case. And how do you do it? Well, we have an inner product, and where we can decide what the best projection is to be closest. We have other cases where we do alignments where we use nonlinearity, and I won't go too much into details, but the output in this case is something which is nonlinear, but we can still do uncertainty contours. And again, this uses the fact that you have a distance, and we can find the best consensus, and we can register the points as they occur, in this case, in 3D. And we did a Bayesian model for this alignment, and it worked well. It gives uncertainty point. If you wanted to do a full Bayesian model with factors instead of just one underlying gradient, you can. And so in a paper that appeared in JASA in 2017, we did this kernel generalized factor analysis. And the output, and the student who sent me the data, he said, "It's terrible, it's so noisy, it's really bad." And I said, don't worry, it hasn't been aligned yet. And once we aligned that data, and this is you get a consensus space and you get the projection which aligns them, this is what you get. So it's a very different picture. And it's nice and neat, and you have everything that is on the right. And you can see here we had very big uncertainty contours for 2 or 3 samples. And we went back and looked at those samples, and they had very low depth, so they weren't deeply sequenced, and so that's where the uncertainty comes from. This is the microbiome of the dog. And so I'm going to stop there and let you ask questions, but I wanted to summarize by saying I learned a lot, long time, been working a lot, but I was sort of, when I teach the course that we made with Wolfgang, the Modern Statistics for Modern Biology, I have a homework set, and I made the LLMs do the homework, and they get very good grades. And I say, this is the end for us. Where does a human belong? And it turns out that as I was doing a lot of work using large language models, I came across a lot of mistakes, not the hallucination mistakes. They're mistakes because the literature doesn't contain the messy things like alignment that we do. And so it doesn't know about it. And so there's still place for humans where we know the secrets of how to do a good data workflow. And the first one for me is the root of all evil in statistics is premature summarization. That is, taking ratios or summarizing your data very, very quickly. Don Knuth always says the root of all evil in computer science is premature optimization. And so premature summarization means that you're going to cut corners in order to make your data as neat as possible. That's the main thing that the large language models do. That is, and it's in what you think is noise and you're cleaning up the data, there might be very important information, so you should do it very slowly and not try to jump that step. So, for instance, I see people putting in their summarized experiments the first two principal components, where in some of the data, it's the first two that they need, but in others, they need actually 2 and 3, because those are the ones that align with 1 and 2. And so you have to be very careful in the choices. So if you're going to keep your summarized experiment, keep 15, because you don't know the context and how you're going to relabel them. And we do have a lot of non-identifiabilities that come up, and you can, there are very simple ways. Anchoring and doing comparisons with latent variables are a way to overcome that. And as always, to this community, choices are not forever. We do reproducible workflows as long as you save all the sufficient statistics in the codes, and your analysis is only as good as the explanation and the software to go with it. So, and thanks to my past lab and previous funding from the Gates Foundation, and many wonderful postdocs with whom I've worked, and of course, I benefit from the tools and the schools of many statisticians. I'm thankful to POSIT and to Quarto and to the bioconductor community. And I'd be happy to take any questions.
Okay, great. We have time for a couple questions.
Thank you for the talk. So I just have so as I understand, you were trying to model how the microbe changes over time and so on. And the standard way to do is using negative binomial models, or there are other models. So I was just curious, how does this perform to the other models which people use?
Well, I don't know what you mean by that. The one model which is completely wrong is the compositional data model.
Yeah.
That's wrong. We agree on that. The other models that people use, which is one, for instance, zero-inflated gamma Poisson is very good.
Okay.
And it's just a question of interpretability. And so the topics very often give meaningful communities, assemblies of bacteria, which are working together. And that's why, and it's simpler. And so in some sense, it's a lower dimensional representation. It's probabilistic. And so that's why it's given very good results. And Chris Sankaran, for instance, who has followed this up with several packages for doing that in many situations. So not all microbiomes are created equal. So the vaginal microbiome, there are maybe 50 main taxa. In the gut microbiome, you have probably 3,000 taxa that are playing. And so you need different tools and different ways of simplifying. So not all models work everywhere. I'm just saying, finding the latent structure, sometimes it's not excited. Sometimes the first topic we find is a contamination because people have used a reagent which is contaminated. And that's a very boring topic. But it's still necessary to take that topic and take it out and say, okay, now the contamination is gone and we can carry on. But it is worked pretty well. And usually it's not just one bacteria, it's a set.
Further questions?
Fantastic talk, Susan. I'm still probably stuck on your question before about PCA and making sure that you don't change the scales of plots, which I think it's...
I have a super reference for you. It's very well explained in this book. There's a whole chapter, and in that chapter on PCA, we do the examples of the turtle data. In the turtle data, you have points, and 99% of the variability explained by one axis, which we would call an axis of size, like 99%. So really, those points lie on a line, and everybody, oh, we have to make a square. Why do you have to make a square? No, these are. No, what you want is the distance between the 2 points is as represented on your plot, is as represented as possible between the distance in the space, in the high dimensional space. So that's, it doesn't, if you have a square plot, sometimes if your eigenvalues are very, very close, square plot, very good. But many times people are not paying attention, and they're making. But do read the chapter 7 in that.
So where I wanted to take this was that I think this really speaks to the point you were trying to make, which is that there's things that people do in terms of the whole community, and there's things that there's things that people do versus they should be doing things the right way or even the Susan way.
No, we have a new word for it. It's the human way.
The human way. But this is the point I wanted to make. So LMMs kind of they're probabilistic. They probably converge to what the majority of people do, and people tend to do things the dumb, simple way.
But this is the problem.
Have you prompted it to say,
exactly,
do this the Susan way, and then it still gets it wrong?
Well, I work a lot with the LLMs, and I was telling people, it's like working with a student you can't trust. And so you make them go away and do something, and then you check. And then you make them go away, and you check again, and you keep checking. It's like having a team of 20 students, and so they can actually work really fast. But you learn that you tell them, I want you to use version 4.2, and because they're looking up documents for you, right? They're looking up the documentation of a bioconductor, and they're telling you to use this command. It's the wrong version. And so you have to be like you would be with somebody who doesn't have a lot of experience. And so you have to be very directive. And you can be very, very, it's amazing what you can do, but you have to be very bossy, right?
So is that any different? Is the human way any different to the machine way if you're doing that with students anyway?
Well, I've learned over the years that when they come back and say, oh, it doesn't work, the best way of doing it is say, okay, I'll do a little example. And this is what I do. I do a little example, and then the students say, oh, yeah, I can do it better than that. And this is always. And it's the same. That is, here's a little example. Do it this way.
Okay, I think in the interest of time, we'll close the session here. Let's thank Susan once again.